Hello everyone. So today we are going to learn a new chapter, reproduction in lower and higher plants. Now in this chapter, the first part we are going to learn asexual reproduction, in which fragmentation, budding and spore formation along with few other types we'll be learning. Also we'll be learning vegetative reproduction, cutting, grafting and tissue culture. Also with relation to the topic, few animation images and few beautiful images which you are going to observe with relation to the topic, as always put to make the content or topic interesting. In the first slide you can see two animation image, one towards the left where you can see for example a spore formation or budding, or by binary fission, how the spores or the asexual reproduction takes place. Also on to the right side you can see the artificial method, that is by grafting, how we can carry out reproduction asexually, that is the artificial means. Now moving on to the second slide, here I put again a lot of animation images which help us to understand the different ways in which reproduction takes place in the lower animals. So you can see the first one by fragmentation in Spirogyra, okay, which we will be learning in detail in the further part. Second, budding in yeast. So you can see how budding takes place. Third, spore formation in Chlamydomonas. Fourth, formation of two new cells, that is by binary fission in Paramecium. Fifth, conidia formation in Penicillium. Sixth, gemmules formation in sponges, that is freshwater sponge. And seven, binary fission in bacteria. So these are some of the types, or you can say 7 types, which I have put the animation image for a understanding of how the reproduction takes place in the lower animals. Now moving on to the next slide, where we are going to learn the introduction part of the chapter. Now here the first point, reproduction is the production of young ones like parents. As we know, the reproduction is nothing but from the parent the offspring is formed. So production of young ones which are similar to parents. Second point, reproduction is an essential process as it leads to continuation of species as well as to maintain the continuity of life. Now what does that mean, continuation of species? If the species don't continue, or from the parents the offspring and the offspring the further generation does not take place, after certain period of time the species may get eradicated, okay, and also it helps in the continuation of life. So the this continuity of species helps in continuation of life, and this also lead to further variation, evolution, etc. Each organism has its own particular method of reproduction. That is, every organism has its own way of reproduction, as we saw in the previous slide. That is 7 different ways I had shown, whether it is binary fission, budding, gemmule formation, conidia formation, right? Different ways are there by different organism. Now, all these methods generally fall in 2 categories. So everything can be categorized in 2 ways. One is asexual reproduction and secondly sexual reproduction. So I put an image showing asexual reproduction with few examples. Unicellular, you can see amoeba, how from the mother's cell 2 daughter cells are formed which further divides into 2. Then Hydra, the bud formation takes place, which again starts to grow and then separates from the parent body. Third, sweet potato, how the underground roots are formed. Then sexual reproduction, that is mainly by gamete. So the male gamete and the female gamete, that is from the father and mother, which is diploid by fertilization, forms the zygote. The zygote which grows into the embryo, which is diploid. Moving further into asexual reproduction. As I mentioned, in this particular part of the chapter video, we are going to learn about asexual reproduction in detail. So first point, asexual reproduction does not involve two compatible gametes or sex cells, or as simply means in asexual there is no fusion of gametes or no fusion, so there is no fertilization as such. It takes place by asexual means, right? Which are the means that we'll learn further, and some of the animation as you already seen. So it is a process resulting in the production of genetically identical progeny. Now since the mother cell divides into daughter cells, the progeny form or the offspring form is same to same or identical or a duplicate of the mother from a single organism and inherits the gene of the parent. So even the genes, the characteristic is similar to that of the parent. Such morphologically and genetically identical individuals are called clones. Now as we learn in biotechnology chapter about cloning etc, but here in asexual reproduction, the reproduction which occurs asexually are nothing but the offspring formed as good as clones. Organisms choose to reproduce asexually by different modes or ways. So here again I put some animation images for better understanding. The first one you can see Spirogera, Paramecium, Bursaria and Rotifers. Spirogera, as you can easily identify the long thin structure which has got the green for pigment, that is a chlorophyll, whereas Paramecium you can see the slipper shaped structure, and small you can see conical structures which is the rotifers. Clear? Then here you can see a fungi species burst open to release the spores, thereby carrying out the reproduction. Then third, budding in yeast. So yeast, as you can see, formation of buds takes place and it in continuation forms new species, which is again a asexual mode of reproduction. Now moving further to the three types which are learning. First one, the fragmentation. Now from the name fragmentation, the formation of fragments, small parts, right? Now first point, multicellular organisms can break into one or the other region. Example, Spirogera. These fragments grow into new individuals. So the image which is there helps us to understand better. We can see a single linear Spirogera where nucleus is present, septa is present, spiral chloroplast is present. Now it divides into two fragments or two fragments of form. Further, to see the detailed part of the Spirogera, I had used a label diagram where you can see pyrenoid, nucleus, cell wall, cytoplasm, mucilaginous sheath, the outer part which protects it, chloroplast which helps in photosynthesis, cell membrane, vacuole and cytoplasmic strand. And here I put another image how the Spirogera appears under the microscope. Then moving further, the second type, that is budding. Now in budding, it is the most common method of asexual reproduction in unicellular yeast, right? Again, as you saw the animation image earlier. So here you can see in budding, as the name suggests, formation of buds takes place from the mother body and it continues to form daughter cells. So here usually it takes place during favorable conditions by producing one or more outgrowth, which is nothing but buds. These buds on separation develop into our new individual. So you can see two images. One, as you can see, many outgrowth or the buds forming in the yeast. Second, I put the animation image step by step how the buds are formed in the yeast. So yeast, which has got a central nucleus, the parent yeast cell, where the small outgrowth, that is a bud formation, takes place, which again has the nucleus in the bud and the cytoplasm starts to divide. And the fourth D part, you can see from the parent yeast, the chain of yeast cells form and many daughter yeast cells are being formed. Now third type which we are learning is the spore formation. Now in spore formation, as the name suggests, formation of spores takes place. That is, example in Chlamydomonas, asexual reproduction occurs by flagellated motile zoospores which can grow independently into a new individual. So here you can see the image of Chlamydomonas, how it appears under the microscope. The labeled parts: stigma, which is the eye spot; nucleus, starch grain; pyrenod, which is the starch membrane; cytoplasm, chloroplast, Golgi apparatus, mitochondria and vacuole, and also flagellum, which helps in the movement, right, or locomotion. So you can see in the second image how the Chlamydomonas, from the parent cell, the zoospores are released, and with the help of flagellum they keep moving in the water body as it is a zoosporangium. Okay, so these are the three types. Now other than three types, we are learning few more terms related with the asexual reproduction. That is, other methods of asexual reproduction include binary fission, which occurs in amoeba paramecium. Now all of you have seen amoeba or paramecium, where the parent or the mother cell divides. So you can see here the parent amoeba, the cytoplasm, nucleus which is present, which starts dividing into two and finally divides into two daughter cells by binary fission. Second, conidia formation in penicillium. Now conidia formation you can again see over here, that is penicillium by asexual reproduction, conidiospore, conidia, that is the B part over here, and germinating conidia. So after it gets released, this conidia just starts to germinate. Okay, third, gemule formation in sponges. Now here you can see the gemule where inner membrane, archeocytes, outer membrane, micropyle and monoaxon spicules are present. In a higher image you can see how it actually appears. Okay, so that is the third type, gemule. So other than the three main type of asexual we are learned, here three more we are learning. So remember the name: binary fission, and the example amoeba paramecium, conidia, penicillium, and gemules, that is it takes place in sponges. Now moving further, can you recall? Earlier we also learned the capacity to reproduce by vegetative propagation. Now what does this vegetative propagation means? Any propagation which takes place with the help of vegetative part of the plant, which can be root, stem, leaf, or any part of the plant body which can reproduce, or we can stay as propagate, right? So one which propagates to form a propagation or propagates to form a new species. So here in case of root, sweet potato, aspergillus, dahlia. So you can see the image also I put so as to understand the examples very clearly. Okay, then leaf, bryophyllum, kalanchoe, begonia. Again you can see the leaf of bryophyllum, how from the leaf new buds are formed and it gives rise to new species. Then this begonia, kalanchoe, how it appears or how it looks like, that's why I added the image in the next slide. So you can see all the names in the next slide. Stem, I think this most of you might have seen, that is rhizome, turmeric. Okay, or another example, if you give ginger, again I put the image of the ginger over here. Then tubers, best example being potato. Bulbs, that is onion. So these are very familiar: turmeric, potato and onion. Clear? Now, and in the images we can see guava, sweet potato, dahlia, roots of dahlia and roots of sweet potato. And here ginger, even the labeled parts is there showing internode, scaly leaf, bud, node and the adventitious root. Now, as I put the images of the different vegetative propagation, so which helps to understand much more easily. So you can see here sweet potato, the plant, and the sweet potato root, which is usually underground. Then asparagus. Next, dahlia. Now all these are in case of roots, right? Leaf, that is bryophyllum. Kalanchoy, see how it appears, small flowers in it, and begonia. So you can see the colorful image in this slide. Clear? Now moving on to the next slide, that is vegetative reproduction. Now plants reproduce asexually through their vegetative parts. That's why the term given as vegetative reproduction taking place with the help of vegetative parts of the plant. Okay, then hence the new plants formed are genetically identical to the parents. Now because it is asexual reproduction, the genetic character is identical, or as we used earlier, the exact clone of the parent. There are also few methods which would not occur naturally in plants. So there are some methods which is not natural, we can carry out by artificial means, that which is carried out presently in agriculture and horticulture to exploit vegetative reproduction in order to multiply fresh stock of plants. So by using artificial method in agriculture and horticulture, we can have more reproduction, much more in a faster way, more yield, and new varieties can be obtained. So artificial methods are used to propagate desired varieties according to human requirements. So whatever is our need, accordingly we can use. So there are various methods which are follows. That before moving to that, we can see the animation image. So two animation image I have put how by cutting, grafting, this artificial method can be carried out. Now in the present scenario, we have got various tools available through which cutting can be done easily and grafting can be carried out with much more ease. Now coming to the individual type, first is cutting. The small piece of any vegetative part of a plant having one or more buds is used for propagation. So in case of cutting, one or more buds are used for the propagation. So again, in that case, you have got stem cutting, leaf cutting, root cutting. In case of stem cutting, most common example being rose, and other than that, bougainvillea. Second, leaf cutting, sansevieria. Now sansevieria, we might have not come across the name, but it is commonly seen around us, which is otherwise called a snake plant. So again I put the image in the next slide to understand what is the sansevieria actually. Then root cutting, example blackberry. So here again I have shown the image how the cutting is carried out in different parts. Cutting in case of stem, then here the other image leaf cutting, and then the third image, that is root cutting. Now the images for a better understanding. Here bougainvillea, this also you might have seen, it's like ornamental plants usually seen in gardens and now surrounding. Then second, rose, which is more commonly used in cutting, where you can have different colored species related to rose. Third, blackberry. Then fourth, the sansevieria, or otherwise called as snake plant. Now this particular plant, if you see the leaf is more familiar, the flower is sometimes rare to see. And there is another name for this, that is mother-in-law's tongue. That is for snake plant or sansevieria. Okay. Now second type is grafting. Here, as the name grafting, two different parts are grafted and made into a new species. So first point, here parts of two plants are joined in such a way that they grow as a single plant or one plant. So two parts of the plant is taken and joined. By joining them, new plant species is formed. In this method, part of the stem containing more than one bud, that is scion, is joined onto a rooted plant called stalk, which is called as grafting. So we can see in this two part is that one, that is stalk, which is already rooted into the ground or the soil, and second one, the scion, which may be a bud or part of the stem which may be grafted into this plant. whereas budding is also called as bud grafting. So if you're using a bud, it is a bud grafting. Otherwise, more often we use the stem for the grafting purpose, in which one bud is joined onto the stalk. The example: apple, pear, rose, etc. So here you can see how the grafting takes place. Now that is why in the first animation slide I had shown about the grafting, how it is carried out. Even in grafting there are different methods of grafting, that it may be T-shaped method or crown shape method. So various different methods are present in it. Here you can see a T-shaped cut is made and a root is inserted, or a plant bud is inserted over here. Here one part of the plant, which is the stalk, and the other scion taken from another plant is joined, which is made by giving a slanting cut, joined by a cloth or a polythene sheet, and then it is kept for certain period of time, whereby the conducting tissues gets joined. Now again you can see the examples, that is apple, the scientific name Malus domesticata. You can see in a single plant you can see two different colored apple, one the reddish one, other the green colored one. And similarly you can see here I also put the flower of an apple. Then second image is of pear, scientific name Pyrus species. Here again you can see the pear and the flower of the pear plant. Then third type is tissue culture. Now tissue culture, again from the name, culturing of the tissue, right? So here it is a method by which a small amount of plant tissue is carefully grown to give many plantlets. That is, micropropagation method is also used nowadays. So we say micropropagation of tissue culture. You can see a leaf, the tissue sample scrap from the parent plant is taken, then it is put it into a petri dish. So tissue sample placed in an agar growth medium containing nutrients and oxygen. So in a petri dish where the nutrient medium is present, the oxygen, that is the growth hormone, is there, which helps in the tissue culture. Third, the samples develop into tiny platelets. Now this tiny the plantlets which is present in the plate, or we can say the petri dish, is transferred into a soil which is placed in another rectangular tray. As it starts to grow, you can transfer into a agricultural field as well. So in a small given area, you can cultivate hundreds of such species in a lesser period of time, and very quickly we can have a more yield. Okay. So the assignment question based on today's part of the chapter. Hope it is clear, and the animation image as well as the images helps us to understand much clearly. So thank you all of you. The remaining three parts I'll be putting in the other videos.